Hi everyone, wherever you're listening from, I'm so happy to be sharing my paper with you. I'd like to start by thanking Team Lumiere for giving me this research opportunity. I'd also like to thank my mentor for guiding me with this project. My research is a literature review that deals with two primary questions. First, what evidence exists for the use of leukocyte and platelet-rich fibrin to promote the healing of diabetic foot ulcers? And second, what does this evidence suggest about using LPRF to treat DFUs? Now, right off the bat, there are two complex terms, diabetic foot ulcers and leukocyte and platelet-rich fibrin. But to understand those, we need to start with the big picture. 1 in 10 adults between 20 and 79 years of age have diabetes today. So chances are of the 10 adults you know in your family, one may be living with the disease. But what is more alarming is that 1 in 4 of these adults develop chronic diabetic conditions called diabetic foot ulcers. Now, these foot ulcers are large open wounds on the lower legs that can result in infections so severe that the foot may have to be amputated. So for many years, scientists have been on the lookout for low-cost and easy solutions to these diabetic foot ulcers. And that brings us to LPRF. LPRF is something we call a biomaterial. It is made by centrifuging the human blood at a very high speed. And this causes the blood components and chemicals to separate to form a lump of blood. Now, this lump is essentially a fibrin mesh that is filled with cells and chemicals. And these cells and chemicals have been shown to promote healing. Because of this potential of LPRF to heal chronic wounds, doctors have already tried it in diseases like leprosy. And they have even predicted that LPRF can help heal diabetic foot ulcers. But sadly, the studies and clinical trials done in this regard are very minimal. So my objective with this study was to synthesize everything that I could find with regards to LPRF in treating diabetic foot ulcers. My methodology was fairly simple. I conducted a literature search on PubMed and Google Scholar using carefully chosen keywords. I also searched up articles, clinical reviews, and trials from the reference lists of articles I already had. Then I used a series of screening checklists called critical appraisal checklists and my own inclusion and exclusion criteria to narrow down the number of results. I went from 138 initial search results to just 10 records. Now 10 is a small number for a literature review, but it just goes to show how little work has been done in this field. With summarizing and synthesizing the paper's findings, I looked at the study designs, the LPRF preparation protocols, and the main healing results in the clinical trials. And then I used spreadsheets to tabulate the main findings and then summarized all of them in tables for my paper. I also tried synthesizing the key differences in healing results, LPRF preparation, and the conclusions of each study. My findings were striking. Immediately, I could see that there were no randomized control trials done using LPRF for diabetic foot ulcers. Now, because randomized control trials are considered the highest grade of evidence for any new medical intervention, without these studies, we cannot really be sure if the treatment is effective. The very limited number of studies was one problem, but another common problem I found was the lack of standardization in the preparation protocol for LPRF, which may have led to different compositions of the LPRF clot, yielding different healing rates. Even in the studies themselves, the subjects had generally not been classified according to baseline characteristics, like age, underlying health conditions, and the subjects had various etiologies. Meaning, for example, that if one patient in a study had diabetic foot ulcer because of neuropathy, another patient had diabetic foot ulcer because of a condition called peripheral arterial disease. Now, these differences in the causes of the wound caused inconsistencies across the board. It was also very difficult to compare the results of different clinical studies because they had very different designs. Some were controlled clinical trials, others were uncontrolled studies, and they couldn't simply be compared. Aside from these findings, I also briefly reviewed the biological properties of LPRF and found that the leukocytes or white blood cells were responsible for promoting pain relief by the release of proteinases or enzymes. Similarly, platelets within those clots were responsible for releasing chemicals called growth factors that are important in the wound healing process. The fibrin mesh, which is the protein casing, encapsulated these molecules and enabled the slow release of growth factors and cells over long periods of time to allow steady healing. The slow release also reduced the number of frequent changes and dressings needed. To sum up, I think that LPRF is almost a miracle because it is simple, cost-effective, efficient, and more importantly, it unleashes the power we carry within our own blood. Such technologies are called autologous technologies. And I hope that experts start looking at this more seriously, because clearly not much has been done as of now, when a lot can be done. Thank you so much.